So now discuss class switching. Class switching is a process in which one isotype is converted into another isotype. It all started from the B cell, which are mature, which are just formed from the bone marrow, and they are expressing these are two IgT, as they have a genetic code for IgD and IgM. Already they have genetic code for IgD and IgM, and they want to and this IgG or this IgM, it undergo react from an antigen, and the B cell presented this antigen to the CD T helper cell, and this T helper cell is activated, and it also a signal to or proliferate, releasing cytokine and CD40L to the CD40 receptor of the B cell, and it determines where this determine isotype switching. So this isotype switching happens in the B cell genetic code, which has a code of IgM, mainly IgM, and the cell need IgM to be converted into IgG or IgE or IgA in response to the respective antigen. So let's suppose this antigen is from mucosal membrane giardiasis, or any enteroviral protein or enteroviral antigen, and the body doesn't need IgM. IgM just indicated there is a primary response, but the body needs anti-enteroviral or proteins, entero antigens. They need some they need some protection against the mucosal membrane, so they need IgA. And how this is possible that IgM is converted into IgA, or for example in the TB we need IgG. So this all happens under the process known as class switching, and this happens in the germinal center of the lymph nodes. And in this process we have an enzyme DNA recombinase, which cleave off or splice out the portion, the variable portion of the IgM, and convert the remaining constant portion and add some variable portion of IgG or IgA, and splice out the variable portion of IgM, and now it has let's suppose genetic code for IgA. So this is A, this is the constant region, this code for IgA. So this protein is now this genetic code transcribed and translated into IgA, which is then secreted from the B cell, and then the B cell then this all IgA protein split off. It be a plasma cell. This B cell gets class switched, activated, and then it form plasma cells. These plasma cells secrete the respective AGE immunoglobulins. So this is all the process. There is an MCQs of DNA recombinase and what happens, splicing out, class switching happen in Golgi germinal center, which receptor, CDL receptor, cytokine. These all were the MCQs which more or less came across in my experience. So when this class switching happens, afterward we have a variety of or specificity of immunoglobulins, and we have different immunoglobulins, and then those different immunoglobulins are IgM and IgD, which we have already. The class switch to form IgG, E or AGEH, and we will discuss these immunoglobulin one by one. First we discuss IgD, as it's just a minor immunoglobulin. IgD is just a monomer, and it's present on the B cell surfaces, and in serum nothing, just book lines, nothing new. First we discuss, then we have IgM. The first one, IgM, which happens in the acute response, primary response, primary immune response of our body. It is IgM. We have a rule for all the immunoglobulins. They all exist as monomers with some exceptions, and we will discuss in those categories. So IgM is a primary response, immune response immunoglobulin, and it fixes complement fixation by their Fc portion. Complement, it fixes complement, and it's a pentamer. It in serum it exists as monomer, but when it is secreted it becomes a pentamer with the J chain, and this pentameric structure give IgM 5 binding sites, and because of this multiple binding site, the avidity of IgM increases, because the IgM bind from 5 sites to a single antigen. It just captures that antigen and does not allow it to react, or does not allow it to do any harm to our body. So IgM has multiple epitopes or multiple binding sites. That's why it increases avidity. Avidity means just increase in the avidity is just the strength of all connections. So until IgG or any natural killer cell or any other complementary defense evolve, the IgM come and capture that antigen. This long chain is the antigen, and remain captured it until the immune response is evolved. It secreted as pentamer, although it exists in serum as monomer. It has some J chain, and it has 5 bindings, and it fixes complement for the IFC. It is immune response, primary immune response, and also it is related to cold, autoimmune, hemolytic anemia, section hematology, autoimmune anemia, autoimmune anemia. And after this IgM, we have then the secondary structure, IgG, secondary immunoglobulin. It is the immunoglobulin of secondary response, exists as monomer, does not secrete it. It fixes the complement, and opsonizes bacteria and neutralize any kind of bacteria or viral toxins. Okay, then it is also the main antibody in secondary immune response. It is the most abundant in serum, most abundant autoimmune in serum, and it is the only immunoglobulin which crosses placenta and give the fetus a passive immunity. And G for good, G for warm and hot. It is related to warm autoimmune hemolytic anemia, cold and warm autoimmune anemia. So after IgG we discuss IgA. So this is the most abundant one. This is the most produced one. Produced antibody from the plasma cell. It is inhibit or prevent IgA prevent bacteria or viruses adherence to the mucosa. So it's actually the mucosal protection. And as we have a lot of mucosa, skin epithelial, GIT, scalp, and a lot of mucosa, then that's why a lot of tissue has the mucosa, so a lot of production, but its serum concentration is low serum concentration, because in the serum we don't need any IgA, because uh, the bacteria or the pathogens which are mucosa associated is not very much applicable to the endothelial of the blood and blood vessels. So that's why we don't need IgA in the blood. We need IgA in the structures, in the barriers, in the epithelial mucosal barriers. And this is the IgA which evade or which protect itself, escape or protect itself from luminal proteases. Proteases act on the hinge proteins cysteine and proline and destroy the immunoglobulins, and it protect itself from proteases by taking or covering up the secretory component of epithelium. It is the only immunoglobulin which transcytoses. It came from one side of the epithelium and just all across the whole epithelium, taking up the secretory component, and evolved or secreted from the other part of the epithelial cell and secreted as a dimer with J chain. These are the high yield points for IgA. And for the mucosa we have enterovirals, giardiasis, and if this IgA is deficient, we are susceptible to giardiasis, especially in the B cell diseases known as Bruton or combined variable immunodeficiency. And it is produced from the GI cells, GIT from Peyer's patches, and it is released into the milk, breast milk, into the saliva, into the tear, into the sweat. It releases over there. The next we have IG. We have discussed IgD, we have discussed IgM, IgG, IgA, and the last one is IgE. So regarding IgE, E for eosinophils. IgE exists as a monomer, and it is anti-parasitic. It uses ADCC killing to anti-helminths. It binds to mast cell and basophil to release histamine, which causes a hypersensitivity reaction type 1 immediate, and also it is the immunoglobulin which cross links with allergen or antigen, and we have inflammatory mediator of histamine, and contributes immunity to eosinophil anti-parasitic. So this is all about immunoglobulins, their class switching. Then we have a last thing, Hinge. Hinge is actually cysteine disulfide with proline, and it is susceptible to proteases, and the susceptibility is increased in the hinge proteins of in the immunoglobulins which have no hinge, for example IgM and E, and the IgGAD, they have the proteases, and also they have long proteases, so they do not have any kind of susceptibility to proteases. IgM and IgE has susceptibility proteases. Rather, GAD has long hinges, and this longness of the hinge, it increases the flexibility, it increases the multiple sites for epitope binding sites, and if we have multiple sites, then we have increased avidity. We have a lot of connections, so avidity increases, and this is all about hinge and Ig immunoglobulin and class switching. And I hope I didn't miss any point. So next we will discuss complement pathways.